Ottoline Leyser, who's based at Sainsbury Laboratory at the University of Cambridge, and she is now the director of that institute since 2013. Just to give you a bit of background to Ottoline's career, she started off with both her BA and her PhD at the University of Cambridge, focusing on genetics, and then had a series of postdocs in Indiana, in the United States, as well as Cambridge, before she took up her own position at the University of York, where she worked from 1994 to 2010, setting up her own research group. Her interests are very much aimed at understanding the role of plant hormones in plant developmental plasticity, and in particular focusing on the regulation of shoot branching as a model. And one of the key players we'll hear today is Auxin, but also other hormone residuals that are involved in plant branching into this research. So over her career, Ottoline received many prestigious awards, including the Society for Experimental Biology Presidential Medal in 2000, Royal Society Rosalind Franklin Award in 2007, and more recently the UK Genetic Society Medal in 2016. She's been appointed very recently Dame Commander of the Order of the British Empire in this year's New Year's End Honours List. She's also a Fellow of the Royal Society and one of the few people outside of the US who's actually a member, a former associate member of the US National Academy of Science. So we're really delighted to have her here today to be amongst us and give us a presentation. The title of the seminar is Dual Mode Strigolactone Signalling from the Bud Activation Switch. From up below. The floor is yours.
Thank you very much. Thank you very much for the very kind introduction, and thank you for coming. It's always a pleasure to visit Rothamsted. I've had a lovely morning and I'm looking forward to the rest of the day, and it's great to have this opportunity to tell you something about the research that's been going on in my group for quite a number of years now. We're, as Kim mentioned, I'm really a developmental geneticist by training, and I have always been drawn to plant systems because, unlike animal systems, plants have this extraordinary developmental plasticity. So the same genotype can give rise to an extraordinary range of different phenotypes depending on the environmental conditions in which the plant is growing. So the plant is not only doing all the things that animals normally do, which is this kind of very interesting iterative loop between the information in the genome and the information in the cell and in the organism to drive forward a developmental program to produce something with 2 arms and 2 legs and a head or whatever. But on top of that, the plant is rolling in environmental information to modify that body plan according to the environment in which the plant is growing. And the model that we work on to try to understand this process is the regulation of shoot branching in Arabidopsis, and in particular we use nitrogen supply as a really easy to manipulate environmental variable. And I think this very simple kind of core system illustrates very well the kinds of things I've been talking about. So you can see this Arabidopsis plant here. This is not going to work as a way to point. Do I have another way to point? Is there a pointer? There isn't a pointer. Okay, so I shall do pointing with words. So on the left-hand side, you can see a plant that's been grown under high nitrogen conditions. So Arabidopsis is a rosette plant. It makes this young rosette of leaves, and then it sends up an elongating primary inflorescence. And on high nitrogen conditions, in long days, you see very little branching before that floral transition. And then after the floral transition, the buds activate in a basipetal sequence from the top to the bottom. So every one of the leaves produced by the primary shoot apical meristeme has an axillary bud. That bud can remain dormant or it can grow out, and that activation is regulated here very clearly by a developmental input, namely the position on the plant. So you see this basipetal sequence of bud activation, but also very clearly an environmental input, because if you grow exactly the same genotype on low nitrogen, as you can see on the right there, whilst you maintain this basipetal sequence of bud activation, it stops early. So this you can point with this, good. This node at the bottom of the plant here, of the elongating inflorescence stem anyway, here the bud has decided to be inactive. The one immediately above is active. whereas buds at equivalent nodal positions and indeed nodal positions below in high nitrogen fertilization are active. So you've got individual buds on this plant making yes, no, do I grow, do I not grow decisions based on integrating information about where the bud is in the plant, where it is in this basipetal axis, plus how much nitrogen availability is there. And whilst some people look at this picture and say this is extraordinarily boring, all that's going on is that the plant with low nitrogen hasn't got enough nitrogen to make any more branches, so of course it's not making any branches. This is very clearly wrong, and I'll get back to that later in the talk. What's going on is really quite a complicated investment decision that the plant is making. It has a certain amount of carbon and a certain amount of nitrogen available to it, and it's deciding where to put it. And under low nitrogen conditions, the sensible place to put it is in your root, which is where you're likely to find the additional nitrogen. So it's all about bringing together this information and then making these investment decisions, which have been evolutionarily tuned, presumably to maximize the fitness of the plant, or rather to maximize the number of grandchildren it makes. So we'd really like to understand the networks that underlie this decision-making process in plants, and of particular interest from that point of view, if we get back to the animal comparison, where animals are making these kinds of ongoing decisions about what to do with environmental information, most of that is relayed through a central processing system in your brain. In the plant context, because they can't move and therefore are very vulnerable to predation, any kind of central processing is out of the question, because no part can be unlosable. Plants on the whole have no unique parts. Every single one of them can be eaten and it's fine because they've got another one. And so this decision-making process that I've talked about how to make your investments in carbon and nitrogen in roots and shoots, has to be done in a distributed way rather than in a centralized way. And so how this distributed decision-making happens is very much what I'm interested in. So kind of stepping back a step, the reason that plants are able to tune their growth and development continuously throughout their life cycle is because they grow continuously throughout their life cycle through the action of meristem. So at the tip of the shoot and the tip of the root is groups of stem cells that are churning out the shoot and the root. They're building the shoot and the root. So here is it's a SEM of a tomato, shoot apical meristem from a paper of Chris Coulemeyer's, and here's the group of stem cells that we all know and love that is dividing and pushing cells to the side of the meristem, and on the edge of this dome, the leaves are initiating in this beautiful spiral pattern, and then in the base of every single one of these leaves is a whole nother one of these things. So the shoot system is highly modular, and one can think of these meristems as churning out successive modules, and these have been termed phytomers, and the phytomer you can think of as consisting of a leaf, an axillary meristem associated with that leaf, and a chunk of stem. And because this axillary meristem has exactly the same developmental potential as the primary shoot apical meristem, effectively the shoot is fractal. So each of these axillary meristems is like these, and each one can potentially build a whole nother shoot. So this is where you get this amazing possibility of body plans based on a very simple reiteration of these phytomer modules. And the question about how many times you reiterate these modules, how many modules do you make, how many times do you activate new axillary meristems, is very much the question about how the body plan of the shoot should be. So really the question comes down to how do these axillary meristems decide whether they should remain dormant or whether they should activate to produce a shoot. And this has been investigated for many years. One of the really interesting phenomena that has really become central to the way I like to think about this problem comes from looking at these little two-node systems. So if you take an Arabidopsis plant and you slice out a bit of the elongating inflorescence that has two nodes, so two leaves, two axillary meristems, and you just ask what happens, then even that very, very simple experiment tells you something really quite interesting. So what happens is one of three things on the whole. Either both buds grow equally, or both buds start to grow, but then either the apical bud or the basal bud continues to grow and somehow manages to inhibit the other bud. So, and we know that this is this arrest of the second bud, whether it's the apical one or the basal one, is as a result of being inhibited by the actively growing bud, because if you chop off the actively growing bud, you can reactivate the bud that's stopped. And so we measure the extent to which both buds are growing compared to one bud, whichever one it is growing, using this thing that we call the relative growth index, which is the length of the longest branch divided by the length of both branches. So this is going to vary between 1/2 when both branches are about the same length to close to 1, where one branch is very long and the other branch is very short. And when you plot this statistic out just from this very simple experiment, you see very much a bimodal distribution where the little X plants pretty much either look like this, or they look like this, or they look like this. And this tells us something rather interesting about what's going on, and it's very clear that the interaction between these buds are competitive, and you can have a situation where either both of them manage to win the competition somehow, so they both are growing, or one will manage to inhibit the other. And you can almost see this going on. So if you do this experiment with just one bud, so you only isolate a single node, then that bud will grow out with this kind of kinetic. This is just tracing a single bud. So this is the gain in length day by day. And by day 3, after the excision, it's already in this rapid exponential growth phase. If you have 2 buds, both buds start to elongate, but they're elongating pretty slowly. So by day 3, neither of them are very long in comparison to this single bud. And you can always see them arguing with one another about who gets to go. And sometimes they both go, and then in this particular case, the top bud won and the bottom bud lost. So the bottom bud will stop elongating, and the top bud then disappears off into this rapid elongation phase, but it doesn't make it into this rapid elongation phase until several days after, if there was just one. So this communication between them, this argument, the competition takes time. And this is an important lesson for life. Communication is very important, it takes time. So this means you have to get out of your office, go and visit your neighbour, taking time and communicate, and information will flow, and it's a good thing. And you can, as I say, you can pretty much see this happening in the plant, the buds talking to one another, because it delays the decision making as they swap information about who's in the best position to grow, one, the other, or both. So I'm really intrigued to understand how this communication happens. How do these 2 buds talk to one another, and how do they make that decision? And as a geneticist, the way I've always gone about that, primarily, first and foremost, is to make mutants that don't do it right. And so primarily what I'm going to be talking about today is the story of a set of mutants we recovered when I first set up my lab in York many years ago now. And they are mutants that we called MAX mutants for more axillary growth, illustrated here by MAX1. And what you can see is that these MAX mutants are extremely branchy, and you'll see that they're very branchy on high nitrogen, and they're very branchy on low nitrogen compared to these plants that I've showed you before. So if you take away these genes, you wind up with a plant that activates buds that are not activated in the wild type situation, regardless of nitrogen availability. So I think this is a very powerful indicator that, in fact, both of these plants are not making as many branches as they could possibly make. They are limiting the number of branches they're making. They're making a decision to be kind of conservative about how many branches to have, and that conservatism is at least partly mediated by these MAX genes. We originally recovered mutations in 4 genes that gave this phenotype, and to cut a variety of long stories short, it transpires that these genes are all involved in either the synthesis or the signaling of a plant hormone strigolactone, which looks something like this. And a long time ago, we established that this is indeed a long-range mobile signal in plants that allows this communication to happen between apices and limits the number of buds that activate in a plant. And again, a very simple experiment that I think illustrates this very powerfully is just a grafting experiment. So this is a grafting experiment involving MAX3, which is another gene involved in the biosynthesis of strigolactone. And if you graft a MAX3 shoot onto a MAX3 mutant root, you get this very bushy plant whereas if you look in a wild-type situation, you get a less bushy plant. These plants look a little bit different from the ones I've showed you before because they were grown in short days, which we do to exaggerate the phenotypes because then they make more leaves, and if they make more leaves, they have more buds, and if they have more buds, they can have more branches. So you see a strong difference between the wild-type and the MAX3 mutant. If you take a wild-type shoot and you graft that onto a mutant MAX3 root, then it looks wild-type, which is maybe what you would expect, but maybe what you wouldn't expect is this plant here, where you take a MAX3 mutant shoot and you graft it onto a wild-type root, and it also looks completely like wild-type. So the genotype of this shoot is the same as this, but it's looking completely wild-type because the thing that's missing, which turns out to be strigolactone, can be supplied by the root. And we know now that the root is in fact a major source of strigolactone in the plant, and it's transported upwards, presumably in the transpiration stream, and can modulate in quite a dramatic way shoot branching. And, but I would point out this plant here, if you only can make strigolactone in the shoot, you'll still not branch it. It's not that the root is the only source. The shoot is fine. And in fact, we've done these, John Booker in my lab a long time ago now did these scary interstock grafts where you take just a couple of millimeters of wild-type hypercotyl and you stick it in between a mutant root and a mutant shoot, and even that is enough to give you pretty much completely wild-type branching in the shoot. So this is very potent stuff, the strigolactone made in the plant as a way to modulate shoot branching. So to cut even more long stories short, we now know quite a lot about the strigolactone biosynthesis and signaling pathway. We have focused very much on the signaling pathway. And what we know is that it looks actually very similar to the gibberellin signaling pathway, which is very well known at Rothamsted, obviously, in that there is an alpha-beta hydrolase family member that binds the strigolactone. It's called D14. Unlike the gibberellin receptor, D14 still has catalytic activity, and the best models out there at the moment are that the strigolactone is actually hydrolyzed in the receptor, and one of the products is retained on the receptor, and it's actually that product receptor complex that is the active receptor. And so it's a sort of non-catalytic in that you don't need the amplicative aspect of it to get to the active receptor, but you do need the cleavage activity of this alpha-beta hydrolase. In that active state, it can interact with a small family of proteins that are in the heat shock protein 101 chaperone-like proteins. The rice equivalent is called D53, and this complex interacts with an SCF-type ubiquitin protein ligase, for which the substrate selection subunit is this F-box protein MAX2, and this results in the ubiquitination of the SMAXL protein and its subsequent degradation. So there's an awful lot of evidence now to support this model from the genetics, from biochemistry and cell biology. I'll just show you a couple of things. So we have made tagged versions of a whole bunch of these things, and you can watch this strigolactone mediated kind of complex assembling in this kind of way. So this is results from FRET-FLIM experiments. So if you have a CFP-tagged D14 protein and a YFP-tagged SMAXL7, that's one of these HSP101-like proteins that's in the pathway, they don't interact unless you add the synthetic strigolactone GR24, at which point they interact. The eagle-eyed among you in the audience will notice that the SMAXL7 seems to exist in kind of nuclear speckles, and when you add strigolactone, the D14 protein seems to be recruited into those speckles. We have no idea what that means, but it is a reproducible phenomenon. We also see strigolactone-dependent interaction between D14 and MAX2. So here is CFP-tagged MAX2 and YFP-tagged D14 that don't interact until you add strigolactone. Here the interaction seems to be across the nucleus rather than in these speckles. We cannot detect a direct interaction between MAX2 and SMAXL7, so we imagine which you can see not happening here. So we imagine that the D14 is the bridge somehow between the F-box protein MAX2 and the SMAXIL protein. So we don't see fret between SMAXIL and MAX2, but we can between SMAXIL and D14 and between D14 and MAX2. So that's what we think is going on when strigolactone is added. And we can also see very clearly that this results in the destabilization and degradation of the SMAXIL protein. So here is SMAXIL7 expressed in root tips, and when you add strigolactone, it goes away pretty quickly. It's pretty much all gone within 10 minutes, unless you try to do it in a strigolactone signaling mutant background, like here illustrated with MAX2, in which case it doesn't go away. So this all fits together according to the pathway that I've shown you. And as a geneticist, I'm much more inclined to believe the genetics than any of that stuff. And here's the genetics, which tells you if you make a triple mutant that takes out all of the relevant SMAXILs in this family, you can completely suppress the MAX2 branching phenotype. So here's a wild-type plant. Here is the SMAXIL 678 triple mutant. Here is the very branchy MAX2 mutant. And here is the quadruple mutant, which completely suppresses this high branching phenotype. And here are the branches quantified here. The very branchy MAX2 completely suppressed by taking out 678. So the only job of the strigolactone signaling pathway is essentially to tune the levels of SMAXIL 678 in the cell from the point of view of shoot branching. And the reason that MAX mutants are so bushy is because they can't do that. They can't degrade SMAXIL 6, 7, and 8. And so these proteins overaccumulate at very high levels. And this high branching phenotype is the result of the overaccumulation of these proteins, such that when you take them away, it's completely reverted. I should point out that the SMAXIL 6, 7, and 8 mutant by itself or in combination with MAX2 is still got quite a lot of branches. It's actually barely less branchy than a wild-type plant. And this is an important point that I'll get back to shortly. So really what we'd like to understand then is how these SMAXIL proteins regulate branching. That's what this whole thing's been kind of reduced down to, is the level of SMAXIL proteins have a profound effect on shoot branching. How do they do that? And as my title suggests, there are two models out there for how they work, and as biology always comes up with, the answer is both of those things, and quite likely probably something else we haven't thought of yet. But I've left the something else we haven't thought of out yet so far because we haven't thought of it. But I'll tell you about the two things. The first thing is the kind of easy thing that is kind of one's gut reaction to this kind of pathway. As you'll have noticed from those fluorescent proteins I showed you, this is all going on in the nucleus, and so there's an instant assumption that what's happening is changes in transcription. And there is quite good evidence that changes in transcription are important. So the model for a long time has been that these SMAXIL proteins are acting as transcriptional repressors, very much in the same way, for example, as the AuxIA proteins or the Jazz proteins do in auxin and jasmonic acid signaling. And this hypothesis has been sort of fueled, at least in part, by the fact that these SMAXIL proteins have a conserved ear domain, which allows them to interact with transcriptional co-repressors of the topless family. So the idea, just like the jasmonate and the auxin signaling pathways, is that there is some transcription factor family that we don't really know what it is yet, that is binding to the promoter of strigolactone inducible genes, but these genes are kept off by the SMAXIL proteins interacting with this transcription factor, recruiting topless and shutting down transcription. And the prime candidate gene to be regulated in this way is the BRC1 gene in Arabidopsis, which is the TB1 gene in maize, and this is core gene involved in domestication in maize that works to do a variety of things, one of which is to modulate branching. And so in Arabidopsis, we know that BRC1 is important in branching because when you make a mutant, you get a branchy plant. There is a BRC1 close homologue called BRC2, so typically we've worked with a double mutant just to convince ourselves that we really have got rid of all the relevant BRC activity. This is work from Pilar Cubas's lab that shows us this gene is expressed in axillary buds here, in the base of leaves in Arabidopsis. And this really quite relatively specific expression in buds points to a direct role of BRC1 in inhibiting the activity of those buds. And when you look at BRC1 expression in the relevant mutants, you see the sort of thing that you would like to see. So here is a level of expression in an inactive wild-type bud, which is normalized to 1. In these max mutant buds, max 2 here, the level is very much lower. This figure comes straight out of a paper from David Nelson's lab, where he's looking at signaling from a related pathway that affects SMAX1, so you can ignore this. But it does show you that MAX2 definitely has very low levels of expression of this gene. But importantly, when you take out SMAXIL 6, 7, and 8, you see super high levels of expression. So this provides quite good evidence that the strigolactone signaling pathway acting through MAX2, degrading SMAXIL 6, 7, and 8, is modulating the level of expression of this gene, and this could contribute to the shoot branching phenotype. But this is by no means the end of the story, and in fact, in my opinion, is probably the minor player in the story, although that I don't know, that might just be my bias in the way I think about it. But the important evidence that has led us to realize that there is definitely something else going on comes from a variety of different directions. And one of those directions is the fact that, as I showed you, these SMAXIL 6, 7, and 8 mutants are still actually quite branchy. They still make quite a lot of branches. And even in those branches that are active, they still have super high levels of BRC1 expression. So here are those the two most apical branches on SMAXIL 6, 7, 8 MAX2 mutants compared in length to wild type at the beginning just as at the floral transition, which would be day 0. So the top node, here they are, 2 very long branches, and the next node down, 2 very long branches. And then when you look at BRC1 expression, even in these active branches, it's very, very high. So having very high BRC1 transcription is not enough to inhibit these buds. So just tuning BRC1 expression up and down is not good enough. We also know, as we'll see later, that the BRC1 mutants are not nearly as branchy as the strigolactone mutants. So what that tells us is that BRC1 is neither necessary for bud inhibition, nor is it sufficient for bud inhibition, and it's not absolutely required for strigolactone response. And that's what I'm going to show here. And this to me, is the plot thickens on strigolactone. This is the really interesting part about strigolactone biology that I've been very keen to try to understand in the last decade or so. And we're making progress, but I don't think we've got there yet. So as I spent some time earlier explaining, if you take these little 2-node assays, what you can see is that either you get this 2 buds activating, or 1 bud activates and the other one's inhibited. And you can plot this out in this relative growth index. Now, something very interesting happens to these 2-node assays when you add strigolactone. This is the synthetic strigolactone GR24. We just stick these little 2-node assays in an Eppendorf tube with strigolactone that we feed in through the bottom. And what happens is that the relative growth index shifts from this bimodal split population, some of these and some of these, to essentially all of these and none of these. So the RGI goes up. Now, this tells you already something very interesting, because it is not the case that strigolactone simply inhibits bud activity. Both of these buds are receiving plenty of strigolactone. One of them is not growing, one of them is growing. So what the strigolactone is doing is ramping up the level of competition between these buds so that only one of them can win. I like to compare this to a grants round. This usually works quite well with a scientific audience. You go into the committee, you sit down around the table, you rank order the grants. That's what strigolactone's doing. The budget, that's decided by somebody else. The cutoff line, yeah. I said that exactly the wrong way around. Strigolactone is the budget, not the rank order. So whoever happens to be on top of the list can grow, but because of the level of strigolactone, that's kind of the level of competition in the system, and it is the budget in the grants round. Okay, so strigolactone is not a straightforward branch inhibitor. We know that this is a direct primary strigolactone effect because when you look in a strigolactone signaling mutant here, D14, what you see is a very low relative growth index. So essentially in a strigolactone mutant, everything looks like this and nothing looks like this, and that's true whether or not you add strigolactone. When you look in the BRC1 mutant, what you see is that maybe the relative growth index is a little bit lower to start with, but when you add strigolactone, it responds really quite nicely. So you don't need to have BRC1 to respond to strigolactone by inhibiting your branch outgrowth. But very interestingly, when you look in mutants that are defective in auxin transport, then you see this response. So this is a triple mutant lacking 3 auxin exporters in the PIN family, PIN 3, PIN 4, and PIN 7. They have a very low relative growth index, so really these buds can barely talk to one another. And when you add strigolactone, although you do see a nice shift, it's distinctly below that of wild type. So here we think there are essentially potentially 2 things going on. One might be acting through BRC1, BRC2, and the other might be something to do with auxin transport. So I've thrown auxin transport in here all of a sudden, but actually auxin transport is where we came into this whole thing in the first place. And that is illustrated here. So it's been known for 100 years that auxin is very important in controlling shoot branching. So here is auxin. It's synthesized in young expanding leaves at the shoot apex, and it's actively transported down the plant in the polar auxin transport stream. And these PIN proteins that I just mentioned are an important component of the polar auxin transport stream, although in the context of the polar auxin transport stream, it's not PIN 3, PIN 4, and PIN 7 that people generally talk about, it's PIN 1. So PIN 1 is expressed in xylem parenchyma cells, or accumulates in xylem parenchyma cells, and in vascular cambium. In the main stem, the PIN 1 protein is basally localized strongly in these files of cells, and it's very important in driving the unidirectional transport of auxin from the shoot apex right down to the root in this very well documented polar auxin transport stream. And it's this that we think is really, in many ways, the hub of the regulatory system for shoot branching control and the core of this distributed decision-making process that the plant uses to decide which buds should grow and which buds should not grow. And that comes out of the extraordinary self-organizing properties of the auxin transport network. And our inspiration for thinking about it in these terms came out of the extraordinary and elegant work done by Tzvi Sachs in the 60s, 70s, and 80s in Israel. And what he developed was this concept called auxin transport canalization. And here is the experiment that really got us thinking about auxin transport canalization in the context of shoot branching control. So what he showed was if you take a little bit of pea stem with its existing vasculature here, with the nice strong polar auxin transport taking the auxin down the plant, you make a little notch, you add auxin to the side here, and you go away. When you come back, you'll see vascular tissue differentiation connecting this auxin source to the existing vasculature in the main stem. And he proposed this idea of auxin transport canalization to explain this, where he said if you have an auxin source here that you supplied and an existing auxin sink where you have these basal auxin transporters taking the auxin away down the plant, then an initial passive flux of auxin from this source into the sink is upregulated and polarized in the direction of that initial passive flux. And so you'll then connect the source to the sink, and these files of cells with highly polar auxin transport connecting the source to the sink, some of those will differentiate into vascular tissue. And his arguments to support this are many and various, but illustrated here is one. If you do exactly the same experiment, but you also add auxin to the existing vascular strand, so that now this strand is busy transporting this auxin that's been directly applied, as a sink, it's much less good than in this situation where it's not got any auxin in it. And now, although you have an auxin source, you don't really have that strong auxin sink, and either when you apply auxin laterally nothing happens at all, or if anything happens, it's vascular differentiation that wanders around and doesn't connect up to this existing vascular strand. And this situation, this configuration, is absolutely reminiscent of what's going on in the context of a stem. So we know that auxin is being transported down the plant in the polar auxin transport stream. We know this auxin is extremely important for regulating the activity of this bud, because if you chop the top of the plant and remove the apical auxin source, the bud will activate. If you supply apical auxin, the bud will remain dormant. If you use radio-labeled auxin, the auxin will be transported down the plant. It will inhibit this bud, but it will not go into the bud. But no radio-label to speak of accumulates in the bud. So this auxin here is indirectly somehow inhibiting the activity of this bud, and the auxin transport canalization idea can explain that, because this bud is potentially a very good auxin source. The young leaves, were they to be expanding, would be making an awful lot of auxin that would be exported out of the bud, hopefully into the stem. The stem is a great auxin sink, as I've explained, because of this existing polar auxin transport stream carrying the auxin down to the root. And so positive feedback between auxin flux, between the source and the sink, and pin polarization between the source and the sink would kick in according to canalization, establishing high conductance auxin transport from the bud to the main stem. And if we propose that this is essential for bud activation to allow the bud to kind of get rid of its auxin, and certainly to establish vascular connectivity between the bud and the main stem, then this mechanism alone could explain why auxin moving in the polar auxin transport stream from the apex could indirectly inhibit the activity of this bud. When you chop the top of the plant, this auxin drains away, and that will allow more basal flux from this auxin source out into the sink, triggering this whole canalization process, and off goes the bud. So it's been known for a very long time that there is in fact a very strong correlation, at least, between the ability of this bud to grow and the ability to export auxin. That's been shown by very elegant experiments by Morris, for example. And certainly in our hands, this correlation is clear. And for example, one of the things that happens very early when you chop the top of a plant and watch what's going on to pin polarization in the stem of the bud now is initially when the bud's inactive, you can detect pin protein, but it's not polar. And when you chop the top of the plant, an early event is the polarization of this pin in the bud stem, establishing auxin flow from the bud out into the stem. So that's what we think is happening, but it's quite difficult to get your head round. So we have established a collaboration with Przemek Puszykiewicz at the University of Calgary to model this process. And here's the kind of nuts and bolts of the model. We work at quite a zoomed out, abstracted level where we model a kind of chunk of stem like it was a giant cell, and that chunk of stem has a fair amount of auxin in it, illustrated by the green box. And it has pin protein illustrated by this red line, and it's exporting auxin into the next chunk of stem, and the width of this white band is the flux of auxin from I to K. And canalization at its heart says that if there's a lot of flux, so if there's a lot of this flux, then there will be more pin protein inserted into this membrane, and then of course there'll be more flux, so that drives the positive feedback. And you can capture that in this extremely simple equation, which says that the change in pin protein in the plasma membrane equals these terms, which are the amount of pin protein going in, minus this term, which is the amount of pin protein coming out. So however scared you are of equations, this is not difficult. Pin protein going in, pin protein coming out, change in pin protein. So the pin protein coming out is modeled very straightforwardly as a linear function. So just a proportion of that pin protein, which is mu, is removed every kind of turn. The pin protein going in has a basal insertion rate. That's just the kind of background level. And then this is kind of the nuts and the bolts. This is a Hill function, so it's a sigmoidal relationship between the pin protein going in and flux. So the more flux there is, the more pin protein there goes in, but in this kind of sigmoidal relationship. And it's actually the interaction between the linear removal and the sigmoidal input proportional to flux that makes this system into a bistable switch. And you can watch this in a kind of animation of this model. So here is the root, which is kind of the ultimate sink for all Auxin. Here is a little seedling. That's what it is, really. And what you can see is this chunk of stem. This green dot is its meristem. The meristem will activate and it will make more stem, and each bit of stem will have an axillary bud. We don't bother to measure the leaf, to model the leaf. And here is some basally polarized pin that's going to transport the auxin made in the active apex down to the root. And if I activate it, this is what happens. And what you can see, whoops, let's stop it, is that these segments of stem are being made. You've got this nice canalized auxin transport down to the ultimate sink in the root. When the buds are made, they accumulate auxin, but because there's already a lot of auxin in the stem, the flux between here and here is very low, and that low flux is not enough to drive the positive feedback of canalization because the pins are coming out as fast as they're going into the membrane, and so kind of nothing happens. So effectively, because these guys are made second, and there's already lots of auxin in the stem from the primary apex, and possession is 9/10 of the law, they are inhibited. If we add an extra assumption that when this shoot becomes floral, the amount of auxin it makes drops, and it's no longer making leaves, so that seems reasonable. And for some reason, oops, here we go. Then, when the thing flowers, auxin in the main apex, in the main axis will drain away. When the level of auxin gets sufficiently low to allow flux between here and here to be sufficiently high for canalization, this bud will activate. This bud will then flower, its auxin synthesis will drop, and you will see this successive basipetal activation of buds that is entirely dependent on this canalization process. That's the only thing going on, really. Canalization plus less auxin made in the floral apex gives you this basipetal sequence of bud activation, which in this particular simulation will activate every bud, but all you need to do is organize it that the amount of auxin still produced from these floral apices is tunable, and you can stop that basipetal sequence at whatever point it is. That means there's enough auxin in the primary shoot axis to stop the next bud activating. So that makes this system a very sensitive system for tuning growth and bud activity according to the environment. And indeed, we have some evidence that in the situation of low nitrogen, it's exactly auxin export from each active apex that's tuned, and that's why on low nitrogen, the basipetal sequence stops early. But it's a really nice decision-making system precisely because of the properties I've described. It's a competitive system. It's all about relative things, the relative source strength of the bud for auxin and the relative sink strength of the stem, and it's got these dynamic components because it's also tunable by the dynamics of the positive feedback in that switch. So it generates through what is essentially a system that's based on competition between different auxin sources and for a common auxin sink, and reinforcement, which is the positive feedback in auxin transport canalization. And competition and reinforcement is really a very well-known self-organizing design principle, which, for example, is the thing that rewires your brain. It rewires the synapses in your brain according to how often they're used, according to the environment. So actually, the behavioral principles that allow you as an animal to adapt to your behavior to the environment are pretty much the same that allow the plant to tune its development according to the environment. So this is very nice. It's a model. I like it, as you can tell. But it would be nice if we had more evidence that actually supports it or disproves it, because it's fine if the model is wrong. Hopefully will take us forward into understanding how the system works. So to understand how the system works, we clearly need to understand much better the auxin transport properties in the stem. And as I've told you, auxin is transported down the chute basipetally, and you can follow it by adding a radio-labeled pulse of auxin to the top of the stem. So this is a 10-minute pulse, and then we take this chunk of this kind of 2 and a bit centimeters of stem, and we chop it into little pieces, and we see where the auxin has got to. And if you do that when you simultaneously add an auxin transport inhibitor, then it hasn't got very far. But if you, so that's with after 90 minutes with this auxin transport inhibitor, NPA. But if you don't add the transport inhibitor, after 30 minutes, you've got this kind of nice peak of auxin here, moving at the kind of rate that people give for auxin transport rates, which is about 1 centimetre an hour. And then, interestingly, after 90 minutes, it's very difficult to see this peak. It's much flattened, and after 60 minutes rather, and after 90 minutes, it's pretty much spread all the way out across the stem. And if you wait longer, it will, all of it, get down to the bottom of the stem, but it's not moving in this neat peak. And that tells you that what's going on is something a little bit more complicated dynamically than just one set of parameters carrying the auxin down the stem. And even within the 30 minutes where you think it's behaving like it should, what you've got is this fast-moving auxin that's going more like 3 centimetres an hour, as well as the bulk of auxin that's moving at 1 centimetre an hour. So both of these things say it's not just a unitary system, and the way that this is best modelled is if you've got two sorts of auxin transport in the stem, the highly polar PATS, polar auxin transport system that I've told you about, that's driven by pin 1, plus a much more general, less polar, lower capacity auxin transport system, which a substantial contribution is made up by pin 3, 4, and 7. So we finally got back to pin 3, 4, and 7, which, as you will recall, are necessary for strigolactone response in those two-node systems. So here's pin 1 being expressed in the polar auxin transport stream in this very basal way in xylem, parenchyma, and cambium cells. Pin 3, 4, and 7 are much more broadly expressed, pin 7 particularly so, right across the stem. It's polar but less polar than pin 1. And pin 3, 4, and 7 we now refer to as CAT, connective auxin transport, because they play a really important role in allowing these two buds to talk to one another. Because if you think about it, these two buds, one of them's going to be on this side of the stem, and one of them's going to be on this side of the stem. And if they're exporting auxin into the stem, which I've just tried to convince you they are, and indeed we know they are, that is not going to be a very good communication mechanism, because from this bud, the auxin will go into these vascular bundles, and from this bud, opposite side of the stem, it will go into these vascular bundles. So they're barely talking to each other through the polar auxin transport stream. However, we know that based on pin 3, 4, and 7 biology, there's actually a lot of movement of auxin across the stem, and that cross-stem auxin movement is important to allow those buds to talk to each other, and it's important for the initial flux of auxin between the bud and the stem, because as you will notice, pin 1 isn't expressed in the tissues between the bud and the polar auxin transport stream. So when you take out pins 3, 4, and 7, what you see is slow activation of single buds relative to wild type, and as I showed you previously, this very poor ability of the buds to talk to one another compared to wild type. So if you've got two buds in this relative growth index assay, they can barely talk to one another. Now, what you can see here is that the pin 347 mutant is not entirely happy in life. Pins have quite a lot of other roles, including in gravitropism, but it's a pretty happy mutant, and actually the total number of branches it makes is quite similar to wild type, and this has been a really useful tool in allowing us to understand how the strigolactone pathway regulates shoot branching. And the role of pin 347, as I said, is in this connective auxin transport. And instead of thinking about the auxin as coming out of a bud and going straight into the polar auxin transport stream, actually what's happening is this continuous exchange of auxin between the polar auxin transport stream and these wider tissues in the stem and across and round the stem. So this canalization model I've talked about is really a two-step process, where initially the bud is transporting auxin across this connective auxin transport domain into the PATs, and it's there that the initial polarization and canalization is happening, and you don't actually establish PATs, high capacity polar auxin transport between the bud and the stem until a little bit later in the process. So we've got two-step canalization. So what does this have to do with strigolactone? Apart from the data I've shown you that says pin 3, 4, and 7 are important for strigolactone, we've actually known for quite a long time now that a core function of strigolactone is to regulate the accumulation of pin proteins. And we initially found this in the context of pin 1. So strigolactone mutants overaccumulate PIN1 protein in the stem, and this is completely reversible in the SMAXL 678 triple mutant. If you follow PIN protein on a single cell over time in the stem, what you see is that addition of strigolactone depletes PIN proteins within about 30 or 40 minutes, and you can see that in wild type and in a strigolactone biosynthesis mutant, but not in a strigolactone signaling mutant. And this is resistant to cyclohexamide, a protein synthesis inhibitor. So this is a rapid primary transcription independent response to strigolactone that just happens. PIN proteins come off the membrane. And we've known about this for PIN1 for a long time. We've now looked at PIN 3, 4, and 7, and PIN 7 responds similarly to PIN 1, but PIN 3 and PIN 4 do not. So it's specific to those two pins. And if you take this information and you feed it into that model I showed you earlier, and you model strigolactone as removal of pins from the plasma membrane, and you make an in silico strigolactone mutant, what you see is this. You see a plant that's very branchy, because essentially what's going on is these buds are kind of a hair trigger for canalization, because there's no strigolactone, pins don't come out of the membrane very much. So if you've got a trickle of auxin that's putting pins into the membrane proportional to flux, and those pins are staying there, then the next turn there'll be more flux, and the next turn there'll be more flux, and the next turn there'll be even more. So you haven't got that counterbalance that's taking the pins out of the membrane because you've taken out the strigolactone. So you see very branchy plants which overaccumulate pin protein. You also see that each of these shoots will export more auxin because they've got higher auxin transport capacity, and auxin synthesis is under feedback control, so if you can export more, you make more. And so you see this massive accumulation of auxin down the stem, which we have measured and indeed occurs quite spectacularly in strigolactone mutants. So here we've got a second mode of action for strigolactone. And at the moment, how this works is very speculative, but I've told you that all the action for strigolactone signaling is in the nucleus. So our hypothesis is that these SMAXL proteins, maybe as well as interacting with transcription factors, are interacting with a mysterious purple box protein, which we don't know what it is. And when you add strigolactone, the SMAXLs are degraded, and we argue that this purple protein is being sequestered in the nucleus by the SMAXL protein. So this is kind of the opposite of the very famous NF-kappa beta signaling pathway in animal systems, where proteins are sequestered in the cytoplasm until the hormone comes along and then they can go into the nucleus. Here we're arguing sequestration in the nucleus until the strigolactone comes along and they can move into the cytoplasm and do stuff at the plasma membrane, regulating the endocytosis of the PIN proteins. And we do know that the ability of strigolactones to work can be blocked by leptomycin B, which is a nuclear export inhibitor. So we have some hints that this might be true. So to try to understand this in more detail, what we're now doing is working backwards. We know that some pins can be targeted for endocytosis and some can't. So that suggests to us that there is some signal in the pin that's being identified or being targeted by this pathway. If we can figure out what that sequence is, we might be able to use it to fish out the purple protein, for example. So we're doing these domain swap experiments where we're taking pin 1 and pin 3, and we know pin 3 can't be removed, whereas pin 1 can. And if we take bits of the big intracellular loop of pin 1, which is where most of the interesting regulatory sequences in pins are, and we swap them into pin 3, we are asking which bit of this loop might be able to make pin 3 strigolactone targeted. And I'm running out of time, so to cut a long story short, the answer is this bit. So here is some of the data just quickly. If we've got so here's pin 1, which can be targeted, and pin 3, which can't be targeted. If we've got the whole of the pin 1 loop, then it can be targeted. If we've got the middle bit, did I point to the middle bit? I can't remember. Anyhow, if we've got the middle bit, it can be. And if you've just got the front end, it can't be. So we're narrowing down where in that loop the specific strigolactone regulated sequences are. And we're hoping to use those to fish out the purple protein or other factors that are needed in downstream in the pathway. So now we've got this going on here. We've got a system where the auxin transport canalization system and the transport network and its self-organizing properties are regulating the activity of the buds. And strigolactone is working in two very distinct ways, one of which is to regulate BRC1 expression in the bud, and the other of which is to tune the dynamics of the auxin transport canalization process by making it more difficult for buds to canalize. And this explains why all it does in those two-node systems is increase the level of competition between those buds rather than simply inhibiting, because it's tuning up and down how easy it is to achieve canalization. And if it's difficult, then the buds will take longer talking to each other, and one will win and one will lose. And if it's easy, they'll just do it, and then both of them will win. These two systems we know are largely independent of one another because, for example, PIN34 triple mutant suppresses branching in D14. So here's a D14 mutant, it's very branchy. Here's a quadruple mutant that has 347 in the background as well, massive branch suppression. Here's a BRC1, BRC2 mutant, branchy, but not as branchy as strigolactone, and absolutely nothing happens to it when you take out PIN347. So they're working in very independent ways. We also know that here's a transcriptomics experiment where BRC1 is high in a small inhibited bud. It drops in a wild-type bud as the buds activate. As I already showed you, it's very low in strigolactone mutants. It remains low in strigolactone mutants, even in this PIN347 background, when we know that a substantial part of the phenotype is rescued. So in the interest of time, I'll skip over some data on a broader transcriptomic analysis, which again argues that these BRC1 mutants are doing something distinctly different from what's going on in the PIN347 background. I'm happy to get back to that in questions if people want to know about this. But just to cut to the summary, what we think is going on here is that two things are tuning the activity of buds: BRC1 transcription in the bud and PIN7 and PIN1 accumulation, and therefore auxin transport canalization. This system has this very interesting relative properties. It's all about relative strengths for auxin, source strengths and sink strengths, and involves these dynamic properties. This system is more of a kind of absolute local input, so for example could be tuned by things like light quality, which would be specifically on that bud. Strigolactone is regulating both of them, giving extra robustness to the system, and we think through these two regulatory systems, which have really quite distinct kind of logics to them, distinct properties, that's what it is that's delivering the robust, environmentally sensitive switching for these buds. And we can see that almost in the way that the buds respond. So if you take out the strigolactone and just think about these two systems, what we know about apical auxin application is it will inhibit the activity of buds for a time. So here's a single bud, we're feeding auxin through the top. If we're not feeding auxin, it looks like this. If we add increasing concentrations of auxin, it looks like what's happening is sort of affecting the rate of growth of the bud. That is actually not what's going on. What's going on is we're extending the length of time for which the bud's inhibited. So you can see that very easily with this 1 micromolar treatment. These guys, what's going on is many of the buds are not activating at all. Where the buds do activate, they activate with exactly the same kinetic as a wild-type plant. And you can see that in this graph when you line them all up to the day on which they activated. So all auxin does is extend that time when the bud is thinking about it. And this creates a very strong switching kind of system, which comes inherently out of the way canalization works. There's a lot of hysteresis in the system. And so once a bud is going, you can't stop it again. And we've known that for a long time. If you do exactly those apical auxin application experiments with a bud that's already growing, that's it. Nothing happens. And you can see that here. These are buds that were big at the start of the experiment, and they are uninhibitable by apical auxin. If you look at exactly this experiment, only in the BRC1 mutant, you see almost the opposite. What you see is a very clear dose dependence for auxin. So what BRC1 is doing is adding some kind of robustness to that switching process. If you take out BRC1, the bud can activate, but then it's just not very convinced about it. And in fact, we've seen these buds in these experiments start to go and then stop again. So this dual mode signaling definitely, according to these data, creates a robustness to the system, which is allowing the plant then to tune very carefully and robustly the decision of each bud to activate or not activate, depending on this combination of different inputs. So it's the regulatory logic in this system and its dynamic properties that I think are key to understanding how this distributed decision-making works in plants and how they manage to tune their growth and development according to the environment successfully. And that's where I'd like to stop. And for some terrible reason, the computer has decided to crash exactly at the moment when I can show you the picture of all the people who actually did the work, which is terrible. Here we go. Can you get them? Here they are in the Sainsbury Laboratory, which is very near the train station in Cambridge. So if you're ever in Cambridge, come through on your way. Thank you very much.
Thank you very much, Ottoline, for that excellent presentation, and really shows the power of genetics and a thorough understanding of chemistry and
Thank you.
cell biology. So, questions for Ottoline? In the middle.
That's kind of what's there, don't you?
Mm-hmm. This is
Okay, righto. We're waiting for a microphone, righto.
Presumably there must be lots of variation in how conservative plants are in their response. So from your talk, I would gather that's more likely to be in the Strigolactone pathway than the Auxin setting level of competition.
So we have actually done quite a lot of work on, so these people work on diversity and plasticity. So we've done a big screen of natural variation for end responsiveness in branching. And there is a lot of variation. The genetic basis for it is complex, and we have some evidence that strigolactone is involved in that, but at the moment it's weak. So we haven't managed to link directly any polymorphism underlying any of our quantitative traits, but there is one which I'm moderately convinced by that's over max 3. But we recover in those kinds of QTL type approaches quite a low proportion of the variation, and we think there's it's lots of loci of small effect plus multiple different alleles at each locus, which is confounding our ability to get down to exactly which genes are involved. But you're right, there is certainly a lot of variation.
Right, okay, sorry. That was really super.
Thank you.
The regulatory process could, as you just heard in your answer, be affected by the biosynthesis of strigolactone. What is it in this case? Is there any kind of change, or is it really just this sort of production in the root and then it's the translational flow, or is there some, can you see any changes in the RNA profile of the...
In the case of so for nitrogen, which is what we mostly work on, it's quite controversial. We can't see any convincing change in the transcription of the biosynthetic genes. There's much stronger evidence for phosphate. So strigolactone also has a very important role in rhizosphere signaling. So it's called strigolactone because, as again well known here, it's kicked out into the soil as and it's the thing that triggers germination in striga, which is a parasitic weed. And so then you say, why on earth do plants bother kicking it out into the soil if it just attracts parasites? And that's because it also attracts symbionts, so it's important in mycorrhization. So the synthesis levels in the roots go up a lot in phosphate starvation. They also seem to go up in rice in nitrate starvation, but there's some controversy as to whether that's a direct or an indirect effect. Obviously in Arabidopsis there's no mycorrhization, and although we can see clear evidence for an important role in phosphate homeostasis, the total amount made in Arabidopsis, even in the roots, is actually relatively low. So proper biochemistry in Arabidopsis has been horrible. And I'm not even a biochemist, so I'm not even going there. So it's very difficult to say exactly what's going up, when and where, and in fact even which strigolactones there are in Arabidopsis is a matter of some controversy. So the real answer to your question, I would say, awaits a better chemist than I.
Thank you.
Andy, over there?
Shall we just shout?
No, I think because of the our distant audience.
You can see from the strigolactone mutants that they're also semi-dwarf as well as highly branched. Does anything know about how strigolactone affects elongation growth?
That's a very interesting question. So there are excellent groups working on strigolactone in a number of different systems, principally petunia, rice, and pea. So Catherine Rameau's group in France has done some very nice work that strongly suggests that it's not gibberellin. So that's probably the part of the question that you were interested in. What it is less clear. So whether there's really a direct effect, whether by then it is some more straightforward nutritional effect because you're spreading your growth out over a wider range of branches, it's unclear, and it's a very interesting question, I think. So what we do know, for example, is those various stems are much thinner than in wild type, and some of that seems to be to do with cambial development and activity. And in Arabidopsis, at least, the biomass of those shoots is not very much higher than a wild type plant. So it's the same amount of carbon that's being kind of spread out more thinly. And so it's plausible that the dwarfism is an indirect effect. But I don't really like indirect nutritional effects because I think plants are cleverer than that.
Okay, well, if I might ask you one, you've said a lot about auxin transport being involved, but I wonder what about the communication that's ongoing with plasmodesmata? Is there any evidence to suggest that there's also some fine-tuning involving the plasmodesmata?
So that's another interesting question. D14, the strigolactone receptor, has been shown to move. In Arabidopsis, there's really very little evidence that that's functionally important. There's perhaps a little bit more in rice that that's the case, and there's some kind of correlative evidence in pea that it might be relevant. So there's that aspect to it. And then the other interesting aspect is that there's some evidence, again, mostly from pea, that redistribution in photostimulates when you chop the top off the plant has an impact on very early events in bud activation, and that's been linked to sucrose, which is possible. But I do think it's very interesting there might be phloem-derived signals in buds that are part of this kind of competition process, which, and as I say with D14, there's some evidence that it moves over quite long distances in the phloem, but there could be all kinds of other things in the phloem that are moving. So I think that's always something worth keeping in mind, and a lot of that delivery would be plasmodesmatally based.
Do we have any questions from Northwick? We've not seen you today. No, okay.
Then I think I'd therefore like to thank Ottoline for presenting, being here today, and